In the last two labs, we have introduced and reviewed basic concepts of Unix, including how to use the terminal or command line, how to run basic commands from the terminal, such as list the contents of a directory or make a new directory, and how to create a simple script by combining these different commands into a text file. Using these concepts and skills, we will now begin to dive into methods for analyzing brain imaging data. Specifically, the purpose of this first lab is to introduce you to basic but very common concepts and analysis methods used for structural neuroimaging data. We will introduce you to the FimBrib software library, or FSL software package, and show you how to use various tools from this package to run different processing steps on example MRI data. Specifically, we'll cover how to convert images from the DICOM type, which comes from the MRI scanner, to the NIfTY format, which is a standard format used by neuroimaging packages. Remove the skull from brain images, which is also known as brain extraction or skull stripping. How to correct for bias field inhomogeneities. How to register two images to one another. How to segment brain images. And how to perform statistics on a set of images. We will also go over other useful tools and commands that FSL has to offer and that we will use throughout this lab and in other labs. From the course box drive, you will find 5 example datasets labeled test_subject 1 through 5. Each of these subjects contains a T1 weighted image that was acquired from the MRI scanner. The objectives of this lab are to perform each of the following steps for every subject. For this lab, your assignment is to complete these steps and write up a brief report that includes an introduction, methods, results, and discussion. In general, your lab report should be approximately 1 to 3 pages with additional pages for figures. These reports can be submitted through the course's Canvas site. We have also posted a couple of examples of well-written lab reports from previous semesters of the course to help guide you on what we're looking for. Before jumping into the lab, it will be helpful to review some basic terminology and that we can use to describe the brain imaging data. Recall from the previous lectures the 3 different viewing planes, namely axial or transverse, coronal, and sagittal. Each of these viewing planes provides a different perspective of the data, with the axial plane corresponding to viewing a subject from the top down, the coronal viewing as if we're viewing a subject or a patient from the front, and the sagittal plane corresponding to viewing the patient from the side. But what is an image exactly? Really, an image is simply a matrix of different pixels that have different values or intensities. In reality, because our pixels in our MR images have units of length, and because our MR images are 3-dimensional, each pixel is actually a volume element, or voxel, as seen here in the green box. These individual elements, we will refer to individual pixels as voxels throughout the course. To see how voxels make up images, we can zoom into this T1 weighted image, and you will notice that the image begins to look more pixelated. These individual elements that you see are the individual voxels, and each voxel has an associated number or value, also an intensity assigned to it. So images are made up of thousands and even millions of voxels. For example, if the dimensions of our image seen here are 256 by 256 by 160, then our image is made up of approximately 10.5 million individual voxels, each one of those voxels corresponding to an individual data point. To process and analyze this amount of information, we need to have specialized software. This is where various specially developed neuroimaging software suites and tools come in. There are many different packages available, including SPM, FSL, AFNI, and many others. In this course, we will primarily focus on using FSL and AFNI, which have a wide array of tools for processing many types of neuroimaging data. We will also introduce other imaging packages that are more focused on specific neuroimaging applications, like the MR-Trix package for diffusion brain imaging. Note, while FSL and AFNI are two suites of tools commonly used, these are not the only ones available, and many of the exercises that we perform throughout the labs can also be done with other tools and approaches. So let's introduce you to FSL. FSL, which stands for the Finbrid Software Library, is an extensive set of analysis tools, methods, and pipelines for processing all types of brain imaging data. The package is open source and can be freely downloaded through their website and works across all the major operating systems, for example, Linux, Mac OS, and Windows. It was developed by the Oxford Center for Functional MRI of the Brain, and these tools can either be run using both a GUI or graphic user interface and or the command line, which we will primarily focus on in this course. As I stated, this runs both on Windows, Unix, Linux, and Mac OS. It also requires X11, which allows you to visualize and forward your screens. FSL has both a graphics user interface and a collection of command line or batch type programs that allow you to efficiently process your data. FSL also contains a viewer, which we will use throughout the course to visualize the data in 3 different planes and in time. There are 2 different types of viewers actually, FSL View, which is an older viewer, and a more recent viewer called FSL Eyes, which we will use in the course. FSL also allows you to overlay information in color on top of an image so that you can see either various different statistical images or other types of aspects that we will be looking at in the lab. In this lab, we will use the FSL, and you will use FSL and become familiar with some of these available tools. Some of those tools consist of FSL Math, which is a powerful library of image processing functions for performing simple mathematical operations on images, such as addition, subtraction, multiplication. It can also be used to threshold or smooth data, and the commands can be concatenated so that multiple functions can be applied to an image in a single command. FSL Merge is a method to concatenate 2D or 3D brain images into a 3D or 4D file. This can ease the processing of multiple data sets, and it's sometimes necessary to merge data sets like this for performing image statistics, like when we use FSL's general linear model tool, Randomize. FSL Split splits 4D and 3D files into 3D and 2D images, respectively. This can be used to separate data in different ways and can be useful when wanting to look at the data in these different ways. FSL Info provides basic information about an image file, such as image dimension, data type, pixel dimension, and file type. FSL Mean TS prints average time series intensities to the screen or saves to a file, and it takes the average. The average is taken over all voxels either within the mask that you specify or all voxels in the image if no mask is specified. FSL Stats, which is similar to FSL Mean TS, provides similar functionality but offers more flexibility for calculating simple image statistics. For example, calculating the mean, standard deviation, the maximum across images, the minimum across images, etc. Type FSL Stats into the terminal to see usage in different options. So let's begin using FSL for image registration, image segmentation, and other useful tools. When we use FSL, we will be using FSL primarily from the terminal. The majority of these commands can be called from the terminal, and you will become familiar with their use from the terminal. One nice feature that FSL offers is that if you simply type in a command into the terminal and press enter, you will see a list of different options that become available when using that specific tool. For example, if you try typing BET or FAST or FNIR and FSL maths into the terminal, what do you see? You should see a list of different options that, one, tell you how to use the specific command. For example, the command will tell you the specific inputs and outputs that are required. And it should also give you a list of different options and different functions that this command can do. The power of using the terminal with FSL comes from being able to write scripts in order to automate processing of multiple images rather than having to run each of these steps by hand. We will provide a small example of this at the end of the lab. The purpose of this lab is to introduce you to the basic steps for performing analysis on neuroimaging data. During this lab, you will become familiar with the basic tools that FSL has to offer. You will learn how to convert images from DICOM to NIFTY. You will learn how to perform bias field correction, brain extraction, image registration, image segmentation, and other image statistics, and performing basic image statistics. To get started, let's first download the files from the course box drive to a folder on your virtual machine for the lab. You should find that this data consists of 5 different data sets named test_subject 1 through 5. It also consists of a file called mni_template.nai.gz, which is the MNI standard space template we will use for trying out the image registration. To continue this lab, we will begin by discussing some basic image types.